All right, so let's take a look at what the answers are. So when we look at this passage, Mr. King and I, we really didn't spend 2 or 3 minutes reading through and understanding every single detail of the experiment. We went to the questions and then pulled out the information that we needed. So with number 13, based on experiment 1, at a concentration of 10 millimoles, which drug was most effective at killing bacteria? So we're looking at concentration, and we're looking at millimoles right here at 10. And as we go down, we can look at the number of colonies that are remaining, because we're probably starting with around 50. And as you look, it's very easy to see. Letter R is going to have the fewest number of colonies. So in your answers, answer A is your best option.
14. Based on the results of experiment 3, which drug enters bacteria cells most quickly? So this is where I did kind of have to pull out a little information from the text here. And it talks about this thing called permeability coefficient, measures a drug's ability to break through the cell. Okay, so that's what we're looking for. It says the larger the coefficient, the faster it's able to pass the membrane. So basically I'm looking for which of the drugs had the highest, the biggest permeability coefficient. Now, Mr. Brace, I believe that there's just straight up a typo here. I think we're going to edit it on your copy, but just know that on ours it says 10 to the negative 5th. That really should say 10 to the negative 9th. So I'm just looking through here and trying to figure out which of these is the largest. Be careful, because you're going to see 11 and think, oh, well, 11 is bigger than 7, but these are negative exponents on your scientific notation. That's basically how many decimal places you have. So a number with 11 decimal places is actually smaller than a number with only 7 decimal places. And that's going to make drug R, which is answer F, the correct answer.
Number 15, in experiment 2, if experiment 2 were repeated with drug U and an incubation time of 3 hours, the number of colonies counted would most likely be. So if we are looking at experiment 2, and we're looking at 3. So we don't have the time of 3 hours there. We are interpolating. We're basically looking at the data that's there before and after and trying to figure out what is going to be in the middle. So if we're looking at 3, and the drug we're looking at is U, we're looking somewhere between 41 and 18. And if you look at your possible answers, there's just that range that they give you, letter C, between 18 and 41.
And sometimes they won't give you a range. They'll just ask you to pick an answer, and you just pick the one you think makes the most sense. So again, you might just say, okay, well, this is 40, and this is about 20. What's half between 40 and 20? Maybe around 30 is what you would see. Now, they didn't do that here, but they do that on many questions. So just know sometimes you actually have to pick a number and just kind of take a good guess at what you think it should be. And there should only be one answer that's really reasonable. All right, next question is number 16. It says, which of the following statements best describes the relationship between molecular mass and the permeability coefficient of the drugs as shown in experiment 3? So back to experiment 3. Molecular mass is here. You'll notice that as you go down the table, it increases. And then we also know the permeability decreases. Now, the confusing part is here they kind of flip it backwards. So here we're saying, well, as the molecular mass decreases, the permeability coefficient increases. And that's the correct answer, which is F.
Number 17, which of the following statements best describes the relationship between incubation time and the number of live bacteria in experiment 2? So looking at experiment 2, we're looking at the incubation time. We can see that incubation time obviously increases, and it would make sense that if you're soaking these bacteria in some kind of antibiotic, probably the longer it's in there, the more colonies that are going to be dead. If we look at all the data, that's what exactly holds true. So as we look through our possible answers, A, as incubation time increases, the number of live bacteria increases only. We can eliminate that. Letter B, as incubation time increases, the number of live bacteria decreases only. That's probably our best answer. Let's check C and D just in case. As incubation time increases, the number of live bacteria quickly increases, then slowly decreases. That's not the case. And D, as incubation time increases, the number of live bacteria quickly decreases, then slowly decrease. And if we look at number 17, our best answer according to the data is going to be B.
And last, we are on, are we on 18 or 19?
We're on 18.
Oh, okay, never mind. Not last. Penultimately, in experiment 1, this is 18, what was the relationship between drug concentration and the drug's effectiveness in killing penicillin-resistant bacteria? So back to experiment 1. It says, what was the relationship between drug concentration, okay, so you see it's increasing across here, and the drug's effectiveness in killing penicillin-resistant bacteria, okay? Well, it's pretty obvious that they decrease. All of them decrease here. So it seems like they do decrease as they go across. Now, if you look at the questions here, you'll notice that in H it says all 5 drugs were most effective at the highest concentration used. That seems to be true. We can go through the others, but we're running a little short on time, so just that is the correct answer. And once you see that all 5 drugs were most effective at the highest concentration, logically that kind of makes sense, but just again, on the numbers, it's absolutely true. They were the most effective.
And number 19, the experimental procedures used in experiment 1 and 2 differed in that experiment 1. So we can actually scroll down to, this is where you need to make sure you do a little bit of reading. You need to understand that in this case, experiment 2 specifically says what the exception is. So bacteria were handled as described in experiment 1 with 2 exceptions. All drugs were tested at the same concentration. And then the second part, the incubation time of each cultured was varied. So that's experiment 2. So that would we infer from our answers in number 19 that the opposite is going to be true. Drugs are going to be tested at different concentrations, and the incubation time is going to be the same. So that leaves us with the answer A.
And I missed that question because when I did it the first time, I was thinking about what was different about experiment 2. So I read that, and I just picked the answer that said that. But again, you got to read the questions carefully. I missed that because I didn't read carefully enough where it said what was different about experiment 1. If this is what's different about experiment 2, then I have to go backwards and see what was different about experiment 1. So there is a little bit about data analysis. Hopefully that was somewhat helpful, and hopefully you did well, and this was useful for you in the future. Thanks for your attention. So, Mr. Brace, I heard you were reading a book about anti-gravity.
Yeah, I couldn't put it down.